All right then, so we're very pleased today to have Dr. Oliver Harrison. He received joining us to speak about induction and regulation of commensal specific B cells. And he received his master's in pharmacology degree from the University of Bath and a PhD in immunology from the University of Oxford prior to conducting research with Dr. Yasmin Belkad at the National Institutes of Health. Since late 2019, he's been a principal investigator at the Benaroya Research Institute in Seattle. Studies in his lab revolve around understanding the mechanisms controlling host microbe interaction at the barrier tissues, primarily in the skin and the gut. These two barriers represent the major portals of pathogen exposure and are key anatomical sites for development of inflammatory disorders. The lab investigates the molecular mechanisms of how T and B cell responses to commensal microbes are mounted and function. The goal is to understand how these immune cells promote barrier tissue integrity and repair, and understand how this goes already during diseases. Welcome. Oliver, thank you so much for joining us. Sorry, just letting a few more people in. And if people can, feel free to add questions during the chat. We'll take those at the end of the talk. So dive right in.
Excellent. Okay, well, first, thank you for having me and inviting me to the Sugar Science and to talk about our work, which, whilst not immediately type 1 diabetes focused, I think has some interesting implications and is building some new approaches that may be of interest to the audience. So I hope that's the case. And so I'll tell you what we've been doing, which is, as you described, really trying to understand how we can track cognate responses to the microbiota that resides in the gut, and why we think that's important for understanding how the immune system works writ large. And so there we go. Okay, so I think everybody at this point in life is probably familiar with the concept of a commensal microbiota. These are good microbes that reside within and upon barrier surfaces. They're consisting largely of bacteria, but also of fungi, viruses, and archaea. And these are essential for many aspects of our physiology. And so through now over a century of work using either notobiotic, also germ-free mice, or actually studies of dysbiosis and the changes in composition of this community, the microbiota itself has been implicated in influencing things through host metabolism, actually more recently behavioral and neuronal development. It influences the efficacy and efficiency of things as powerful as cancer immunotherapy and immune checkpoint blockades that were awarded with the Nobel Prize a couple of years ago. They're increasingly implicated in the efficacy of vaccination responses. And we think, because as immunologists, this is all mediated by the ability of the microbiota to promote the development and the education, and therefore the function of the immune system. And how it does this both locally, and in our heads that means within the tissues colonized, so within the gut or on the skin, but also how these microbes transmit those signals throughout the systemic immune response, so to distal sites. There's many ways by which this occurs, and we're not going to touch on all of them today. I think a number of metabolites that are being produced are actively modulating both immune and non-immune cells. What we're really interested in actually is this idea that there are induction of cognate or antigen-specific T and B cell responses to certain components of the microbiome. And they actually act as messengers to tune tissues throughout the body. And really, without this access, we've shown that actually wound healing in the skin is impaired. Infection or immunity to infection in the gut is also impaired. And so really, we're trying to understand and extend our strings into understanding the B cell or humoral response to these microbes within the gut. And this is really a new venture for the lab. It's something that we've not really investigated previously. And part of that is because of the lack of tools to do so. And so what I'm going to tell you today is really our approach to generate new ways to track B cells specific for components of commensal microbes, so that we can further kind of conduct mechanistic studies both in mice and hopefully in the future in humans too. So a large kind of overarching background of humoral or B cell immunity in the gut. This is a well-studied axis. We are by far from the first to investigate this. But really, most of the work that's been done in the gut has implicated this response in the context of either infections or in toxin exposure. And so we know a lot about the role of different isotypes of antibodies that are produced by gut B cells, including secretory IgA. And this IgA has really been shown to be essential for neutralization of toxins produced by different bacteria. It's important, therefore, for pathogen elimination as well. But when we think about the microbiota or the microbes that are present all the time, actually this antibody isotype is really important for maintaining this microbiome where it should be. So confining it to the gastrointestinal lumen and away from the epithelial cells of the intestine that make up that single cell barrier between us and the outside world. But more interestingly still, actually, this molecule promotes the diversity or the kind of stasis of the microbiome as being very diverse, made up of a broad swath of community members, which is considered in most instances to be a healthy microbiome. By contrast, actually, very little is known about the roles of IgG isotypes, which is what people more commonly think and measure when you're surveilling systemic immunity. Actually, very nice recent work has implicated these responses under homeostasis as providing a regulatory role within the intestine. And they actually dampen immune activation within the gut. That's actually in contrast to probably older and more established literature investigating the role of IgG responses to commensals in disease. And so this is actually widely known in a number of chronic inflammatory disorders that you mount antibody responses that are detectable in serum against members of the gut microbiota. And so this is true in inflammatory bowel disease. Actually, there is a test against the OMP-C antigen from E. coli that is reasonable in Crohn's disease. But actually, recent work from people at Benaroya and further afield, including Jane Dansker at Sick Kids in Toronto, has demonstrated that there's antibody reactivity elevated over that present in healthy individuals in pre-diabetic children with susceptibility HLE alleles, and that this is also true in rheumatoid arthritis. And so actually we're thinking of this as really kind of an immunological readout, that actually breaking tolerance to your gut microbiota early in life or at the time of disease onset may be more reflective of kind of mechanisms of immune regulation going awry. So what we've been doing is really in the context of health and homeostasis, but we're really keen to expand our tools, and I'll tell you about those now. And so when we entered the field, really we were struck by the use of two major technologies to ask questions about humoral immunity to gut microbes. And these are both very elegant and very powerful approaches, but they lack one key ability, which is to go in and ask about particular antigens or microbes de novo. And so these approaches, either IgA or IgG-seq, really rely on the fact that gut microbes are endogenously coated with antibodies. And so those antibodies are actually present in stool and can be used to essentially immunoprecipitate immunogenic microbes. And then using 16S ribosomal sequencing, you can essentially see what is invoking antibody responses at the level of a single bacteria. And so that's super helpful, right? It tells you what's immunogenic in the gut. And actually you can do that over time to see how immunogenicity or targeting of different microbes is changing.
Just a second, Oliver, I just wanted to ask you for this slide, is this technique being used in any autoimmune sort of diagnosis or biomarker setting that you know of?
Not of which I'm aware. Certainly it's very heavily used in research context, but I don't think it's progressed through into kind of more clinical biomarker studies as yet.
Right.
I think that's because it's still a little finicky, to be quite honest. I think there is absolutely room for this sort of thing, but I think to make them kind of like clear tests and kind of super reproducible over time, there's still a lot of work to be done, and people are doing that work for sure.
Great, thanks for that.
Yeah. So IgA tells you in a snapshot in time what microbes are being bound by antibodies. It doesn't tell you anything about the B cells that produce those antibodies or indeed where those B cells are. The alternative approach that people are taking is really to isolate B cells from defined gut compartments or from the gut-associated lymphoid tissues, so like the Peyer's patches and the mesenteric lymph nodes that drain the gut, and then take those B cells, sequence their B cell receptors, and then reconstruct those to generate monoclonal antibodies. But then there's a whole kind of downstream slew of having to back calculate and figure out what those antibodies are specific to, some of which are components of the microbiome, some of which remain unknown. We wanted to kind of find a happy middle ground of this, where we can go in and pick out microbes of interest and then be able to track immune responses to those microbes in vivo. And so to do that, we turned to using a model system in the mouse, which is this very well described kind of prototypic commensal microbe segmented filamentous bacteria. And this is a really interesting commensal microbe. It's very immunogenic in that it colonizes particularly within the murine ileum, or the terminal part of the small intestine. And there it induces cognate T cell responses against itself, and really was a main driver in the discovery of Th17 cells within the gut. And so Dan Littman's group has been very nicely shown that actually colonization with this microbe elicits both Th17 as well as T follicular helper, all those CD4 T cells that help B cells. And other groups have really implicated the induction of IgA upon colonization with this microbe. And we think that's because it's really actually immunogenic, because it adheres directly to the epithelium. On the right-hand side of the screen, what I'm showing you is the model system by which people work with this, because whilst SFB, as I will kind of paraphrase to, is culturable, it's incredibly difficult to culture. And so people rely on the fact that actually commercially available mouse strains from 2 different vendors have a differential colonization of SFB. And so on the left, you can see a scanning electron micrograph of the villi of the small intestine from a mouse from JAX, or Jackson Laboratories, and these do not contain SFB. Whereas the mice on the right are from Taconic Farms, and these are constitutively colonized with SFB. And you can see that real filamentous structure binding to the intestinal epithelium. And so we're able to use the difference between these 2 strains, as well as adoptive transfer or fecal microbiome transplant from a positive to a negative mouse, to induce immunity and look at this response. So that's our model system. SFB is a particularly interesting microbe because it actually can protect against infection with its colonization, can protect against infection with other microbes and pathogens, but it also alters and influences the severity of a number of models of autoimmunity, including EAE and UAE, so models of multiple sclerosis and uveitis. And so we set about using this microbe to build tools, really. And to do that, we wanted to be able to track antigen-specific B cells. And that's not really been done before, at least for commensals. So we turned to this experimental system for antigen discovery, which is phage display. So phage display is a really powerful and high throughput approach for finding basically protein-protein interaction partners or protein macromolecule interaction partners. In this case, we're using protein antibody interaction, using antibodies that are elicited either through in the serum or through actually the GI or the gastrointestinal gut wash. And so those antibodies that are really in the lumen of the intestine. And so to do this, we isolated genomic DNA from SFB, sonicated and blunt cloned this into a phage-mid library, essentially an expression library for making bacteriophage. And bacteriophage are viruses that infect bacteria. And what's really nice about phage display is this requirement for genetic complementation. And so we can fuse our antigens of interest into an external protein that's displayed on the surface of the phage, this P3 protein. It's incredibly accepting of introduction of new proteins. And this can be done in this phage-mid vector. And so really the genetic complementation, when we infect our hyperphage and our E. coli library together, essentially means that we only generate phage that are displaying antigens or potential antigens that are in frame, in the right orientation with no stop codons. And so this really enriches for kind of an open reading frame or orpheome. And so from this approach, we generated a library expressing SFB antigens in the region of like 50 million independent antigens and epitopes. And then from that, we took this phage display library and essentially go through a process of what's known as biopanning, which again is using essentially immunoprecipitation using serum or gut wash antibodies to pull down immunogenic proteins derived from SFB. So we use anti-IgA or anti-IgG to then capture serum immunoglobulin or gut wash antibodies from mice colonized with SFB. Over that, we basically bind our library of potential antigens or immunogenic proteins. And then from there, we wash that library off to really enrich the phage that are being bound by antibody. We do that 4 times to be stringent and increase the affinity of these interactions to get really good binders. And from there, we pick individual colonies to make monoclonal phage. So this is to be able to isolate individual phage and individual antigens. And from there, we're able to sequence that phagemid or plasmid to be able to identify those immunogenic proteins. So when we do this, what we can see essentially as we go through our hits, and these are just kind of representative proteins from the SFB genome, in red are highlighted now not only proteins, but really epitopes within these proteins that are targeted by either serum or gut wash antibodies. And so really we can learn lessons from this alone. We can see which proteins are being targeted and even which domains. And we're still working to try and understand if there are kind of broader rules at play for what is immunogenic. But what I'm going to tell you about today really is this immune responses elicited by this protein SFB3340. It's detected both by IgA and IgG responses in the gut. And that was important for us. We wanted to be able to compare and contrast how these responses are mounted. And so what's really interesting about this protein is that it's actually part of how the SFB attaches to the epithelium. And so we think that in part makes it particularly immunogenic. And we wanted to study that, that protein in this regard because we know it's eliciting a very strong immune response in the gut. To do that, we started with kind of well-trodden technologies, again, using this differential colonization between mice from Jackson Laboratories and Taconic Farms. In the top, I'm just showing you ELISA dilution curves against this recombinant protein, and this is very specifically detectable within mice that are colonized on Taconic Farms with SFB, but not those that aren't. We can also stimulate B cells from different tissues with this antigen to elicit production of either IgA, shown here, or IgG, and that's true for a range of tissues in colonized mice, including the bone marrow, mesenteric lymph node, Peyer's patch, spleen, and the small intestinal lamina propria itself. So this was really like a very big step forward for us. This was the first antigen defined for B cell responses in the gut against commensal microbes. But it still kind of falls short for what we wanted because it doesn't allow you to either isolate the B cells directly, you have to in vitro culture them, or again, the serum ELISA is not really related to what's making this antibody response. So we turned to an approach used by a lot of B cell immunologists in the world of studying immunity to pathogens, and that is the generation of B cell tetramers, which is essentially a fancy way of saying fluorescently tagged proteins. What we do is take our antigen of interest, in this case SFB3340, and conjugate it round a streptavidin PE core. And then that is basically a staining reagent that you can use for flow cytometry to be able to pull out antigen-specific cells from the whole polyclonal repertoire and then ask about their phenotype and function. Actually, what's at least as important, and probably more, is the generation of a decoy reagent. And so this is actually to deal with the fact that there are B cells in a naive repertoire that will bind structural components of a tetramer, including PE itself, but things like streptavidin and His tags. And so we generate this essentially with a different fluorescent dye and without the antigen, but with all the structural components. And that lets us pull apart the true, like genuinely antigen-specific cells from background. And then we can, if we choose, use magnets to essentially enrich this population. But from what I'm going to show you today, actually that's not required. All of these are just staining the tissues and cells, and the flow cytometry plots that I'll walk you through. And so when we do this and we take this and what this data actually looks like, on these flow cytometry plots, we can see we're now gating on total B cells and plotting the decoy reagent, which actually picks up quite a remarkable number of cells, against SFB3340 antigen. And whilst there's very few dots in these plots, what you can see is that when we have mice that are colonized with SFB, the frequency and number of these cells actually goes up. There are cells in a mouse that has never seen SFB. And actually, when we find those are all entirely naive. And so that means that for immunologists, they're expressing high levels of IgD and IgM on their cell surface. But really what it means is this is the population that would respond were the mouse to be colonized or see this antigen. It's just never seen it yet. By contrast, if we look in the mice that are now colonized from Taconic farms, actually, the vast majority of these cells have now downregulated IgD and IgM from the cell surface, have kind of switched their immune isotype to either IgA or IgG. And actually, when we gate further on these through the expression of CD38 and FAS, these are all pretty much uniquely embroiled in this germinal center response. And the germinal center is present after, it's a kind of a macro structure that's present after immunization or infection. And it's the site where this kind of Darwinian evolution of antibody affinity occurs. Actually, these structures are present all the time within gut-associated lymphoid tissue. And this is because of the microbiota. There is essentially a constant source of non-self antigen that these B cells are responding to. And actually, in this context, actually now these cells responding to SFB are embroiled in this response all the time.
Oliver, quick question in the chat from
Yeah.
Boston. The Alternus Lab, Kiati Girder, is there a need to for barcoded gDNA to determine which sequence is acting as an antigen? How did you determine the conformation of antigens? And can you use this technique for multiple gut microbes?
Good questions. So I think there were three hidden in there.
Yes, the first one is there a need for barcoded gDNA to determine which sequence is acting as an antigen?
No, there is not, because the DNA that comes through in the phage mid, we can essentially just PCR amplify and then blast against the genome, and then that is the epitope for the immunogen lies within there. We have not done anything to further fine map where those antibodies are binding or if there are multiple binding sites in there. Well, it's something that we've considered. We haven't considered it too strongly for this protein because it's enormous and doesn't express very well at the best of times. But yes, you could do it.
And the second was, how did you determine the conformation of antigens?
We have done nothing to confirm or investigate the conformation of these antigens at all. We've just re-expressed them as recombinant proteins from in E. coli. I will say that we've done a number of these, and some are great and re-express fine, so we assume that they are taking on their native structure. Others we do not and cannot express for reasons that are beyond my little immunologist brain, and so those kind of run into a dead end, and that is certainly a limitation of the system, for sure. And then
the last, okay, and then the last one is, can you use this technique for multiple gut microbes?
Absolutely, and there is no reason why you cannot use your microbe of interest. We are certainly working on 2 or 3 other microbes and trying to find even more clear definitions of what induces IgA versus IgG. It's also not in any way limited to bacterial components of the microbiome. As long as it has genomic DNA and you can clone it into a phage mid library, you can test for immunogenicity. People have been doing this for pathogens for quite a long time. This is just one of the first forays, we think, into commensal microbes. So yeah, you can potentially run multiple tetramers at the same time as well, if you can get your color combinations to work for both tetramer and decoy reagents. Yeah.
Great. Okay, she said thank you.
Thank you. Okay, I am not going to show you data, but you hopefully believe me when I tell you that SFB is sufficient to elicit this response. We've done this in germ-free systems. This does not need other members of the microbiota. And as you would expect from those that know about germinal centers, this is entirely dependent upon T and T follicular helper cells. So we kind of crossed our T's and dotted our I's there. What this now allows you to do, though, because you can actually find these B cells ex vivo and isolate them away from the polyclonal population, is we start being able to ask questions that we couldn't before about the anatomical compartmentalization of these responses. And so for that, for us, actually beautiful work from Simon Milling and then Daria Esterhazy and Daniel Masida have been looking at different gut draining lymph nodes, draining different parts of the intestine and how they induce different affected T cell responses in those sites. And we had a similar question, which was like, okay, is this also true for B cells in the intestine? Are they mounting different responses to the same antigen in different tissues or different subsites? For us, we very crudely just dissect the intestine down into three major subparts: the duodenum or the proximal region, the jejunum, the middle, and then the ileum, which is the terminal part of the small intestine. And then take the Peyer's patches or these inductive sites. Essentially, think of this as a kind of a lymph node embedded in the small intestinal wall. And then assay the response to SFB across these sites. And what you can see actually was really interesting to us, is that the frequency of these cells embroiled in germinal centers seems to be about the same along the length of the intestine. But when we look for the ability of these B cells to induce different antibody isotypes and effector function, actually now we see a dichotomy along the length of the intestine. And so we see very little induction of IgG1 across the length of the intestine. Actually, up in the duodenum or the proximal part, we see very high induction, if not unique induction in some cases, of IgG2B. And that flips when we go to the ileum. At the expense of IgG2B, these isotypes, these B cells are now producing primarily IgA. So this was actually the first demonstration that even within Peyer's patches of the small intestine, there is a difference in the ability of these B cells to respond. And we're really interested in using this as a system to understand more about what drives a B cell in vivo and not in a dish to become IgA or IgG producing, with the idea that actually this is really important for mucosal immunology and mucosal vaccines in general, as we want to try and promote more IgA responses for things like respiratory vaccines. But that's very much hand-waving and very much work still in progress. What we're able to also do is really demonstrate actually something that's been very well kind of understood but never directly demonstrated against single individual antigens, is that when we compare the Peyer's patches, which is kind of like the first line of defense and inductive sites in the gut, compared to the mesenteric lymph node, actually the majority of this responses are listed in the Peyer's patch, but the quality is also heavily different. When we look at the Peyer's patch cells particularly, they're really high producers of IgA in general. There is absolutely no IgA produced against this in the mesenteric lymph node. Why is that important? Why should you care? Well, I think it's really a very striking demonstration of the fact that actually the mesenteric lymph node is the secondary site of drainage for the gut, and actually induction of IgA here is not particularly useful. If you've got antigens translocating from the gut into the mesenteric lymph node, now actually opsonizing, or the ability to kind of bind, complement, and destroy these bacteria is going to be much more useful. And so here actually we see a demonstration that actually only IgG2B is being produced against these antigens, telling you that not only is there kind of spatial control or anatomical control along the length of the intestine, but also levels of defense to stop these antigens kind of permeating through into systemic tissues. Okay. The other thing that we're particularly interested in is really the early life response to the microbiome. A lot of people are becoming extremely interested in this, and I think it's very important because it really sets the tone for how your immune system responds to these microbes throughout life. And this is particularly true in the context of in the context of type 1 diabetes, where pre-diabetic kids are already making altered responses to commensal microbes. Whether that has anything to do with the onset and severity of disease, I have no idea. But I think it's an interesting observation, and it's one that we're really interested in an experimental setting. And so what I'm going to show you here really is the idea that actually bugs are colonized vertically, and so that means that they're transmitted from mum to pup. And that happens early in life, as you might expect. And so in this instance, and I'm sorry, this one doesn't have labels. In white, we've bred mice that do not have SFB together, and we're now looking at the pups. And in gray, we've bred mice that do have SFB together, and we're looking at their pups. And so when we look in the poop of these pups at 2 weeks of age using qPCR to try and detect the abundance of SFB, actually SFB is present in neither of these strains. But by the time we get to 3 weeks, and this is where mice are weaned, they're converted onto solid food and off breast milk, now they're beginning to get colonized with SFB. And by the time they're 4 weeks of age, this is essentially at the levels of adult colonization. So this is a very rapid exposure to new antigens in the gut. At the same time, we're able to detect, and this kind of makes sense, that this response elicited by SFB and tracking these SFB-specific B cells in the Peyer's patches really kind of coincides with colonization. We don't see any at 2 or 3 weeks, but once the mice have been colonized for a week, actually now we're getting induction of B cell responses in the pups. What kind of threw us for a loop until we actually went back to the literature was that you can actually already detect antibody responses to this antigen in mice that are 2 weeks old. And this is before they're ever colonized and before they have any of their own endogenous B cell activation. And what we figured out was actually this is derived in utero from mum. And so this is really very much kind of a demonstration of this transgenerational immunity that's really important for setting the stage for colonization by the microbiota. And here you can see in mice that are born to SFB positive mothers, actually the transmission of both IgG1 and IgG2b into the serum of the pups. IgA is not, although we suspect it's probably transmitted through breast milk and into the gut lumen, but IgA is not taken back up into systemic circulation. And so what this allows us now to do is really to go in and ask questions about early life immunity to gut microbes, but really to now be able to the first time pull apart responses elicited by mum and transmitted versus de novo responses elicited in the pups. So we're really interested in how that induction of immunity in early life is set up and then alters infection and inflammation later in life as well. Okay, and so finally, what I've been telling you about really so far has been very much the description of this kind of natural inheritance of the microbiome from mum to pup. But we also wanted to understand how this response occurs de novo in the context where we actually introduce this microbe into adult mice. And this is a really a very useful experimental system for being able to track and perturb B cell responses in the gut in general. And so one of the things that we'd done was use this fecal microbiome transplant or gavage of poop, basically, and then track this response over time. And really to kind of oh, I think I'm missing a slide. Anyway, to kind of give you the long story short, essentially this looks exactly like if you inherit SFB from mum. You get the exact same germinal center response, and this comes up by about 3 weeks of colonization. And so our real final question for investigating this was to really try to see if we can transcriptionally profile this response better to understand what's going on in that germinal center. And that sounds kind of like a daft question when you think about how much we know about the biology of germinal centers in context of immunization, vaccination, or infection, which is really the drive to promote affinity of these antibodies through the subsequent accumulation of somatic hypermutations within the B cell receptor or the antibody locus that drives increased binding. But this question was never really quite so clear in the gut. A lot of people had kind of hypothesized that the purpose of germinal centers in the GI tract was actually to broaden specificity, and so actually increase the reactivity to different microbes at the expense of affinity. And so now we could actually directly address this question. And so we did that by colonizing mice with SFB, and after 21 days or 3 weeks, pulling out antigen-specific B cells using our fluorescent tetramers so that we're now only studying antigen-specific responses, and then using single-cell RNA and VDJ or B cell receptor sequencing, really start to assess the quality of the immune response that we're getting. And so this is really just a visual representation of some UMAPs that don't really tell you very much other than we can identify the populations we find by FACS, which is those naive B cells that are really not engaged in the response. that germinal center response that's really what we're interested in, and asking whether it's broadening or focusing specificity, and then a small population of antibody secreting cells, which we think are going off to be plasma cells elsewhere. And so what we did was really take these single cells and the amplified VDJ or B cell receptor sequences for both the heavy and the light chain of the antibody, and reconstructed these phylogenetic evolutions of this B cell receptor. And so for a single example, let's say this one VH153 on the left, in blue, we've highlighted basically the unmutated ancestor or the germline sequence for the antibody. And we basically back calculated that by understanding how these somatic hypermutations are picked up through time. And so we've done that for multiple B cell receptor lineages that are present in multiple mice to really map how these mutations either contribute to broadening or focusing of the response. So we generated those monoclonal antibodies that I'm pulling out in kind of colored nodes, and tested their affinity for the recombinant protein SFB3340, either using ELISA or biolayer infrared, I can never say this thing, BLI, essentially surface plasmon resonance. And what you can see in both of these approaches that agree very well is that as these antibodies acquire a number of mutations, the majority of them actually increase their affinity for their target. And what I'm not showing you here is that they actually don't bind in a polyreactive manner to some non-related antigens or closely related antigens. So irrelevant antigens, sorry. So we think that in this context, really, this is the first demonstration of an antigen-specific focusing within the GI tract. Kind of trying to address the question that arose earlier in a very indirect way, we wanted to see if these antibodies actually work in situ where their antigen is being found. And so to that, we turned to confocal microscopy, took one of these highly matured antibodies, and essentially used this to now go in and restain for SFB in situ. And so what you can see in these confocal microscope images, blue is DAPI, which is nuclei, EpCAM in green is marking intestinal epithelial cells, and really the red is antibodies raised against this SFB3340 protein. And this is exactly where we think SFB should be sitting, which is on the tips of the villi and the terminal ileum, where they're kind of really evoking this immune response in tissue. Okay, so this is where we're at now. And just to surmise, I guess, what we've been working on, and then a little taste of where we're going with this. We've been using phage display as a very powerful means to identify immunogenic antigens within the gut microbiota. Again, we focused on a bacterium. Actually, this can be extended to many bacteria and non-bacterial components of the microbiome. What I was talking to Monica about before we came online was actually, this is, I think, a really helpful tool as we start to think about immunity to microbes in different disease contexts. And that's because actually this tool is now no longer MHC restricted, as many tools that have been generated for MHC or HLA tetramers are. And so hopefully we can kind of bounce across species to investigate immunity to microbes outside of the mouse. And now we're really interested in saying, okay, we know how this works under homeostasis, that's all well and good. What happens when you either introduce genetic perturbations or environmental perturbations? How does that influence this immunity, both in the short and the long term? And that's work that we're going to be building on in coming years. Specifically, and I hope, this was the work and will be the work of Dr. Sheena Verma. She's a postdoc in the lab. She's a fantastic postdoc in the lab. And she had a lot of help from other folks in the lab at Benaroya Research Institute as well as locally within Seattle from the UW and Fred Hutch. And we've got some fun collaborations going on with the other folks around the world. And I'd just take 1 minute to thank our funders for supporting this. And if there are any questions, I'm happy to try and answer them.
Thank you. This is a really interesting new tool, and the fact that it is non-MHC restricted, so you can use it in other models, is really exciting. I'd like to open it up to the audience. I think the Altinus lab is well represented. Maybe they have some questions. Okay, if not, I have some questions.
Sounds good.
Okay, oops, I'm sort of cut off here. Yeah, so as at onset of type 1 diabetes stage 3, children present with, and others, I guess, present with a significant decrease in diversity of the microbiome. And so, when you think about that, you think about, okay, are those, why might those ones that are disappearing disappear? Sort of a large global question. And, you wonder, okay, is it some kind of epitope that they display that the immune system recognizes and takes them out? Some kind of cross, recognition mistake?
Yeah.
Or is is there something else, and sort of given your work in context, what are your thoughts on that?
That is a great question. We are not experts in analysis of community composition by any means, but we are certainly getting interested in this idea of either molecular mimicry or cross-reactivity, and how those responses that are elicited actually feed back onto the microbe themselves to influence either it's just colonization levels and quantity, but also transcriptionally what that's doing to the microbe itself. There's some beautiful work out there showing that actually antibody binding to E. coli influences the biology of the bug. So yeah, we're beginning to try and set up to see if, not for SFB, but for other systems, if there are potentially ways we can look for cross-reactivity of autoantibodies and defined known autoantibodies against components of the microbiome, this molecular mimicry. I think that would be an enormous step just to understand if there is any reactivity or cross-reactivity, and then I guess further understanding if that's contributing to diversity or loss of particular microbes. I think will be a whole nother body of work. But yeah, I think it's a really interesting question, right? There's like however many potential foreign antigens encoded in the microbiome plus post-translational modifications. I think it's gonna be a lot of work to get that system working to see if there is, but then I think there's potential for really getting to some of that kind of understanding of, yes, changes in microbiome are one thing, but how does that alter the interaction and the quality of the immune response elicited locally? Whether that relates to disease, again, huge question, but we're trying to make those first steps to look for cross-reactivity at least.
Yeah, and I mean, that's great. I mean, they do have, there's some clues, B. adolescentis is one of the first to go. And, so what are the qualities around that? And it seems like your system might be very, agile in kind of like investigating some of those questions. I wondered also, you mentioned early off camera, you were at ICMI in Copenhagen in July, that meeting. And was there any buzz around this topic as pertaining to autoimmunity or type 1?
Good question. Sorry, I misspoke. My student was at ICMI, but
oh, that's still
fully filled in. Yeah, I think there is. There's a lot of people using the nod system to really now. I mean, the very striking observation of difference in incidence in diabetes in the nod mouse in the presence or absence of the microbiota has been well known and well described by many people, and the sex differences are also recapitulated in that context, right? But I think people are now really going from those first kind of very obvious findings to now fine mapping and really some very impressive perturbation and mechanistic work in those settings to really start to figure if there are particular species or isolates that are influencing that, and more so, what is the functional capability of the microbes that are changing through those contexts, right? And whether that is cognated and T and B cells, or whether it's many of the metabolic mediators that are produced. I think there's some really good people working on that. We are not, but I think it's in safe hands, and really kind of stepping up the game of what has been kind of reasonably descriptive for a while into some very powerful kind of mechanistic studies to perturb immune microbiome and microbiome host interactions. Yeah.
I guess one last thing I would ask for your system. Did you, I mean, you looked at the 3 sections of the small intestine, but did you look into the sort of the large intestine, or have you sort of like ventured into that area? Have you looked into the vestigial, appendix region. There's been some sort of smoking guns around the appendix and what might be happening there.
Yeah, good question. Actually, a really nice paper came out probably within a week of us publishing, saying there are 5 parts of the small intestine, and the 3-part dogma's dead, so we need to go back and redo that. But we, not for this microbe because it is so specific to the small intestine, but we have found different microbes both in the large intestine and ones that live between the two, that we're really starting to answer that question. And no, but I liked the question about the appendix. I think it's a smoking gun in a couple of disease contexts that we're really interested in. So not as yet, but we are trying to extend beyond the GI tract and look for that systemic immune response that I kind of talked about, including kind of within different adipose depots and bone marrow as well. So, yeah.
Wow, you have a lot of work going on there. It's exciting times. Are you looking for postdocs?
Always. I think everybody is always looking for clever people that are interested. So yes.
Okay, great. Well, we'll put that shout out on this presentation, and hopefully, you'll get some inquiries. Last call for questions. Anyone in the audience?
I have a quick question.
Oh, yes. Hi, Emma.
Hi, everyone. Hi, Oliver. I am Emrah from
Hi.
Boston College. So I was a bit late to the talk. I apologize for this, but I think we need more studies like yours to understand host microbe interactions specific to the gut commensals, and we don't have many. So this is one of the pioneers for B cells. My question is, we are very interested in molecular mimicry mechanism, but we are very T cell centric and we are very T cell epitope centric when we think about molecular mimicry. I'm curious about your thoughts whether some gut microbes might stimulate B cells, and there might be some cross reactivity that might start autoimmunity.
It's a great question. Yeah, we I will tell you that we are not B cell experts. This is our first foray into B cells, so we're still trying to figure out what can be done, and the ability to kind of find epitopes for our phage display system. I don't know of any context where there are cross-reactive antibodies, but that is probably more my ignorance than it is the lack of knowledge in the field, to tell you the truth. So I think it's definitely something we need to get into, but I don't have a good answer for you, and I'm not going to pretend that I do.
Thank you.
Yeah.
Well, I mean, if I think if the question's coming from the Ultimus lab, then it kind of shows that there's a knowledge gap that needs to be addressed. So I think it's really it's something that people should think about.
Yeah, certainly we will. Yeah, maybe in touch to pick your brain and see what we can do.
Another great B cell person is, or BCR person, is Stephanie Hanna at Cardiff. Just to drop her name in there. So, okay, then it was a really interesting talk, great techniques, and thank you so much for sharing. Really appreciate it.
My pleasure. Thanks for the invite. Appreciate it too.
Yeah, have a great rest of the day. Bye-bye now.
Bye.
Bye.